In 2014, time flies, the University of Wyoming hired Dr. Holly Ernest as its Wyoming Excellence Chair in Disease Ecology. And they also hired her to head up its Wildlife Genomics and Disease Ecology Lab. Her training in research has led her to describe herself as, quote, a wildlife biologist, population geneticist, and research veterinarian. Now, what does that mean? Wildlife biology. Well, let's take the easy one first. Holly studies Wyoming's wild animals. She looks at the ones we all like to see, the charismatic megafauna, okay? Black bears, mountain lions, bighorn sheep, you name it. She also studies less attractive critters, ticks, bugs, germs. Oh, and by the way, she's an expert on Wyoming's ubiquitous magpies, as well as hummingbirds and owls.
Research veterinarian. Well, from my experience anyway, vets deal with the health and diseases of animals. Dogs, cats, other things. Research means she must study those diseases. But it's the term population geneticist that brings everything home. Dr. Ernest looks at the genetic makeup of and variation within wildlife populations, not just individuals. In particular, she is interested in how genetic variation changes over time, especially under the impact of pressures from diseases and human population. Dr. Ernest earned her Bachelor's of Science from Cornell University, and then in 1986, her Doctorate of Veterinary Medicine from Ohio State, and followed that a few years later with a PhD in ecology with an emphasis on wildlife genetics and population health. She taught at the University of California, Davis for 13 years before coming to UW, where she teaches, in her words, students to grow their appreciation and talents in the art and science, I like that, art and science, of designing and conducting studies involving DNA, ecology, and population health analysis of free-ranging wildlife. Today, Holly will talk to us about her work in the Jackson region that looks at two of its favorite creatures in her talk, Giving a Hoot About Wildlife Genetics, Enhancing the Survival of Western Wyoming's bighorn sheep and owls. Dr. Ernest.
Thank you, Paul. Thanks very much. So our wildlife live very secret lives. In Wyoming, we may think we see what they're doing, but we really see a tiny proportion of what they do, where they nest, how their babies turn out when they reproduce, how disease and predation affects them. So this is really important for conservation. And I have a few things that are at my heart. One is using genomic science to learn more about life on Earth. Another thing is teaching the next generation of scientists. And the third thing is applying my science to conservation of wildlife. And with conservation of wildlife, we need to know what a thing is and what it's doing before we can conserve it. If we don't know what the unit, the species, the population is, we cannot effectively conserve it. So that's a little bit of what I'm going to talk to you today, is to introduce you to this science of wildlife genetics, a few terms. It is a language. And so I'm introducing a few terms about it. I'm retaking Spanish, so I'm very in tune with how hard it is to relearn a language. And then to show you how it's applied. I'm a very applied scientist in conservation, and I picked out two of your species, my species too, that my students and I are working on. So I'll first start with the great gray owl. This is a pretty awesome creature. How many have seen one around this area? Most of you. Isn't that awesome? So that is really exciting. And right here in Jackson is the heart of its habitat. I did want to show you its range. It's not just in Jackson. It's actually through, I'm using the pointer to point at the green area that's on the map. It's circumboreal. So around the globe, in through northern Europe, in through Siberia, there's great gray owls. So they're not just here. So the arrow points to where we are, and you can see it's a little peninsula down here. And I'll also talk about this tiny peninsula of great gray owls that are down through California to introduce this. So they're very tied to their environment, to the boreal forest. That means pine trees mixed with meadows where they can hunt for voles and other small rodents, occasionally other creatures. They're gorgeous animals. They're big. So they're the biggest owl on the planet as far as dimensions. They're not the heaviest owl, but they're the biggest. And they have amazing adaptations for how they survive, which makes them really special. You can see the owl here with the vole over to your left, or sorry, to your right in its mouth. And then the diagram shows you how special it is with its adaptations. It has that facial disc, that flat facial disc that helps hear, just like when you put your hand to your ear, it works the same way to funnel the sound in so it can hear. It's got these amazing eyes and a neck that can turn around, not 360 degrees, but it can turn pretty well. It's got 5 feet of wingspan. It's a huge owl. It's got that dense downy area on its feet and those talons that raptors use so well to hunt with. It's an amazing creature. And you might think that that map I have down here is the same as the great gray owl's range. This is actually a different map of the boreal forest diversity. So this, you can see how similar that owl is tied with the boreal forest. But what you might notice is not much of that forest extends down into our area. You don't see much green down in here in the lower parts below Canada. And so that's something that we think about with conservation of a species that's kind of at its range limits. So I wanted to introduce a few concepts about genetics and conservation as we dive deeper into great gray owls. So when we think about evaluating genetic diversity and how healthy the genetics of a population is and monitoring them, there's a few terms. So one is to think about the genetics of the individual. And this can actually help wildlife conservation through development of tools for forensics to battle poaching of individuals. I'm actually newly appointed to a federal wildlife forensic panel, and I work with forensic geneticists around the world to develop new techniques to make this better. So that's pretty exciting. So defining what a population is what we think about a lot as wildlife ecologists and biologists. As Paul said, I don't know what hat to wear. Sometimes an ecologist, sometimes a geneticist. So I think in all of those terms. And finding out what a great gray owl population is not so easy. We kind of know where they are. We go on eBird, the database that's online where you can post when you see a great gray owl. But that doesn't tell us what a population is. And so I'll describe that a little bit. To delineate units of conservation. So once we define populations of wildlife, then we can define what are called management units or evolutionarily significant units. And one layer of information that's required is genetics. That's definitely becoming more and more important. Determine gene flow. So all of us biological units are just gene carriers, right? We carry our genes, we mate with somebody, and those genes get carried on to the next generation. And so one thing we think about is how those genes flow across the landscape. It's really the animals or plants that are moving their genes, but how does it move? And that's really important. First we have to identify the populations, and then what is the gene flow between those populations? Evaluating genetic diversity. So that's determining whether a species or a population is inbred, or does it have certain genes that are really bad for its adaptation in that environment. Effective population size, I won't go into that too much, is a pretty nerdy thing, but super important. And just as an aside, I have been working for nearly 15 years on a California sea otter project. And one of the things that was identified in the recovery plan for that federally listed endangered sea otter population is effective population size. So when you look at the number of breeding individuals that will go on to the next generation, how many are there? And it turns out that what they thought about 40 years ago is very different from what we can calculate from whole scads of genome data now. So my profession is battling with how to calculate this very important NE, effective population size, that we use in the Endangered Species Act. And another important thing for genetics is how we can look back in time, similar to our previous speaker, but on a smaller time frame, to use contemporary genetics of our species to know what happened in the past. So historical demography means what happened in the past. Was there a population bottleneck, a sudden decrease in population size that whittled their genetics down to a small number? And our genetics now can help give us information about that. That's new. When I started grad school, that wasn't available, so it's pretty exciting. So back to our great gray owls. So there's underneath this bubble is the range in North America. So the greenish is their range. And you can see there's the little peninsulas of populations going down to near where we are in Wyoming and down into California where the southernmost population is. And then most of its population in green is up throughout Canada and into Alaska. So one question we have is the genetics of these owls, is their biology such that they're just one big interbreeding population, panmictic? That's your first terminology word. So panmixia means all interbreeding. And so that's one thing that genetics can help determine. So it turns out their range is actually kind of fragmented. So the red here, and I'll outline it a little bit, describes where they are right now. So here we are in Wyoming, right there, next to Idaho, Oregon, and California. And those of you, I'm sorry if you're red-green colorblind, but I'll outline, there are spaces here between where there's no range of the great gray owl. They are fragmented. So does that panmixia model still work? That's what we want to test. What is a population here? So one idea is you could guess on a map based on their current distribution that maybe the contiguous areas might be all interbreeding or panmictic. Maybe that's one population. Maybe our Wyoming and Idaho populations are one. Maybe the others that are along the Oregon eastern border are one, and similarly more western in Oregon, and then California. So that could be, if we're thinking about hypothesis-driven science, that could be our hypothesis that we could test using genetics that's also layered on the field biology, field telemetry, and other sciences. So one thing with our genetic science that we'll use to test those questions is wildlife genetics is not like when you take a swab from your mouth and send it off to 23andMe or Ancestry.com, where they'll run your DNA and give you some kind of answer for $100. It's not like that with us. So we have to start from the beginning with no prior knowledge and develop our own genetic tools, our own reference databases. So that's what 23andMe, Ancestry.com, the Nat Geo databases do. They've taken millions of people's DNA and developed databases. Without those reference databases and accurate databases, our genetics is crap. We need accurate databases to be able to test things. We need to first develop the tools. So that's what my lab does a ton of, is first developing the wildlife tools, the genetic tools, to even look at these species. So it takes a lot of investment in time and money. And even though people say that genetics is coming down in price, in reality, the price is having the expertise work on it for a long time. Hiring a postdoc or a PhD student who can spend their whole time bioanalyzing this terabytes of genetic data. So that's important to recognize. So back to our great gray owls. So I did come from the University of California at Davis, and when I was there, I started my faculty position in 2002. And in 1999, there was a virus sweeping across from New York City. What was that virus? It kills people, it kills horses. West Nile. So I was there, and it was coming across the country, and I asked myself, what's going to happen to the wildlife species that might be in danger to that West Nile virus? That's kind of how I got my start with some of these species. And I asked myself when I was riding my bike into work, what's going to happen to the Swainson's hawk I see over, the yellow-billed magpie, and the great gray owls? As West Nile swept across, what we discovered was that they were exquisitely sensitive to the virus. If they were exposed to mosquitoes with a virus in it, they almost all died. And the Minnesota Raptor Center suffered that when they lost their whole great gray owl colony, when by mistake it was left outside over a long weekend without mosquito netting. And so I saw that coming. I started to contact the great gray owl biologist. Great gray owls are centered in Yosemite National Park, so I contacted the biologist there and said, what do you want to do? Let's think about what we need to do to find out. And so that was actually the start of my genetics research too, because it turned out they didn't know anything about the genetic diversity of the great gray owls in Yosemite. And so my students and I were hired by the National Park Service, by California Department of Fish and Wildlife to find out some of those issues. So on the map, again, I pointed out Jackson in the yellow arrow just so we could keep track of ourselves. And then this red arrow with this little state of California next to it is where that tiny population of California great gray owls is existing. I'm just going to tell you about them first because I gained the experience with them before I came to Wyoming. So during that time, I was lucky enough to hire this awesome biologist, Josh Hall. Dr. Josh Hall. He's now head of habitat recovery for most of California in the U.S. Fish and Wildlife Service. And so working with him, we developed the first high-resolution DNA panel for these great gray owls and published that work. And along the way, we had to learn how to bleed owls, so we practiced on chickens. The chickens did fine, but I taught with other veterinarians how the crew could take blood samples safely from a great gray owl from its legs. And this actually happens to be Josh's dad, Buzz Hull, who is one of the key people at the Golden Gate Raptor Observatory right near San Francisco. So you have to develop the tools. So the next thing you have to do after you develop those tools is to think about how you're going to use them. I'm going to introduce a few concepts here, and it's an alphabet soup, truly, of too many acronyms, and I'll try not to use that too often. But first, a DNA marker is something that's polymorphic, which means variable, and it can be used as an indicator of some kind of variation. Now we aim for genome-wide variation, so we can tell what's going on across the 6 billion or whatever base pairs each creature has in its genome. And a really important marker for us is a SNP, SNP, single nucleotide polymorphism. So of our alphabet in our DNA, the A, C, G's, and T's that are the chemicals in our DNA, we just have an alphabet of 4 that makes up our DNA, we can have a mutation that happens, and that is a SNP. It's a variation. And there's millions of those SNPs throughout our genome, great gray owl's genome, and we can study that. A really important DNA marker. Another thing is mitochondrial DNA. It's a cytoplasmic DNA. Mitochondria are way cool because way back in ancient times, before all of us animals had mitochondria, the powerhouse of the cell, it was actually an organism that over evolutionary time invaded animals' bodies, brought its DNA with them, and now lives within us as our own DNA. Mitochondrial DNA are the mother lineage. So I passed my mitochondria to my son. My son cannot pass the mitochondrial lineage to his children, but if I had a daughter, she would pass it to her children. It's an awesome DNA marker because it gives us a mother lineage. Very cool. Analogous to cytoplasmic marker is in plants. So they have their own cytoplasmic marker too. And that's a chloroplast marker. A really workhorse for us geneticists is called microsatellites, and they're short regions that are highly variable, and human forensics still uses microsatellites. That's how the O.J. Simpson trial used genetics. All of the forensic trials that you see use genetics in using microsatellites. They're also called STRs and other acronyms. And next-gen sequencing, you may see that, and basically it means using markers across the whole genome. It's a generic term because everything is next-gen this or next-gen that, but just so you know, it just means looking across the entire genome, usually with SNPs. And so a few other terms here, and then I'll go back to the story of the great gray owls, don't worry. So one is allele. So that's when we have like an ACG or T at a certain site in our genome, that variability is called an allele. I might have an A allele, my husband might have a G allele, and my son Graham would have an A/G because he inherits one from each of us. Another is a locus. That's a site in the genome. It could be a SNP, it could be a microsatellite, it could be mitochondrial DNA. It just means a location in the genome. Allele frequency is my currency. So that's what I study all the time. It's how often alleles occur in a population. And so that's really important for genetic diversity. Genetic diversity is that variation in a population or a family. And then gene flow we talked about before, how genes move across between groups, between families or populations. So with microsatellites, I'm just mentioning a few of these again, is they're short motifs. A motif is just a stretch of DNA. So in this case, the microsatellite here is the CA repeat. And what happens is that we have thousands of these in our own genome. Most times we don't think they do anything. There's a few diseases that are microsatellite diseases that are inherited, but they mutate really fast because our mechanism to repair DNA often doesn't see a difference between this 8-segment CA repeat and one that's 9. And so between generations, we can add a CA repeat or take one away, and our repair mechanism in our DNA doesn't pick it up. And that's awesome for us population geneticists because it gives lots of variation in great gray owls or bighorn sheep or whatever we're looking at. It's a great tool. And so again, this is just a bigger diagram to show you how it can change from the 8 repeats to the 9, and then it can mutate back. And this is over thousands of generations. It doesn't happen instantly. But sometimes these mutations then spread throughout a population, and that gives us a tool to test what's going on. The primer part of this is what we use. That's a tool that we use to pick up this microsatellite. We develop DNA from this end and that end, and we put it in machines, and we grab the whole thing bioinformatically. I just wanted to give an example of a tiny data set so they could imagine in terabytes of data what our data might look like. So each row, these are actually mountain lion data. So each row is a single mountain lion. So mountain lions 1 through 5. And then each column is a microsatellite locus, a single microsatellite throughout its genome. And then the numbers refer to how long that microsatellite is. And so you can see in this first locus, it's color-coded. They're all the same, aren't they? So that locus doesn't tell me anything about genetic diversity. It's all the same. Turns out that locus tells me if I pick up a fecal sample and I get this DNA out of the fecal sample, which I have, it can tell me that's a mountain lion. It's not a bobcat. Bobcats have a different one at this specific locus. So that's a handy tool to tell species apart. Now, if we look at the next locus, we can see that this individual is a homozygote. It has the same 2 alleles, whereas this one has a 123, 135. It's a heterozygote. So another part of genetics that's really important is to determine these differences. And if you look at the colors across each mountain lion, they're actually unique. So when we use those in a combination or a haplotype, we can tell individuals apart. The forensic geneticist can tell if that blood sample at the site was the same as the killer, that they're going to try to prosecute. And then we take that data, only lots more of those loci. I only showed you a few columns there. And then we do analyses with it, statistical analyses. Now, this is a color diagram of great gray owls throughout many sites. And if you imagine that each narrow row is a single great gray owl, and then you can tell from the colors that they have different genetic composition. I've color-coded this so that each color means it's a different genetic cluster. So this tells us that their genetics across these sites, from Canada through Oregon and the Sierra, are different. So that was one of the things that helped answer one of the questions. Are they panmictic? Are they all interbreeding? They're not. Now, in this study that Josh Hull and I did, we didn't have Wyoming samples. I wasn't thinking of Wyoming at that time. Now I am. And so that's a question, are the Idaho samples, Idaho great gray owls, right next to us, some of you live in Idaho, are they the same population or are they different? It's important for conservation. We have to know, because if they're unique, we might want to think about protecting each unique population. So that's a question I'll get to in a minute about my grad students' Teton great gray owl studies. But I want to tell you a little bit more about the threats to great gray owls. And so there's more threats such as disease, roadkill, poaching, and Trichomonas is a pathogen. It's a protozoal pathogen that great gray owls get. It's really nasty. It kills them. One interesting thing is that recent studies have indicated that Tyrannosaurus rexes had Trichomonas. There are Trichomonas lesions on T-Rex fossils. And so it's another bit of indication that, yes, birds are dinosaurs. So when I banned hummingbirds here in Jackson, and when I have a hummingbird in my hand, it's a dinosaur. So that's pretty cool. They had the same diseases as our great gray owls. Here's a picture of a roadkill from Yosemite. So great gray owls like to fly low to catch their voles or whatever and right over the road, and then they don't see the car coming. And then West Nile virus. It's something to think about with climate change. We did not find West Nile virus in the great gray owls in Yosemite. One reason is because they are above 7,000 feet, which currently is the limit for the mosquitoes to amplify the West Nile virus in their system. But with climate change and warming in some areas, moisture changing, that will likely change. So that's a changing dynamic. And then of course fires. We've heard, I have colleagues that actually have lost some of their homes in the past week from the fires in California. This rim fire affected the habitat of the great gray owls in Yosemite, and we certainly worry about it here in Wyoming. So those are some of the threats, why it's important to define populations of these animals, and that we use the data from the genetics in concert with other data such as NASA and NOAA geotelemetry stuff from satellites, and also telemetry from the raptor biologists such as at the Teton Raptor Center that study them here. So one question we have for our Teton owls is, are they different from adjacent owls, like in the Idaho? And we suspect that they have probably undergone genetic bottlenecks in the past. And so to study that is one of my graduate students, Beth, who's working with the Teton Raptor Center. She actually worked here for a number of years on the telemetry project with the Teton Raptor Center, and also collaborating with Wyoming Game and Fish. And so she's just developed a whole suite of genomic tools, so whole genome tools, to examine that question. And then she's re-examining the population genetics that Josh and I did in the past. As scientists, we realize sometimes we might be a little wrong, might be majorly wrong. We ask ourselves, okay, can we do better? So that's great gray owls. And to illustrate some more points, I wanted to shift to bighorn sheep. So bighorn sheep are pretty amazing, iconic species. I studied them in California too. We identified genetic interchange across the Mexico-Baja border. That's really important. We're a little scared about the wall because it'll cut off the genetic interchange. That's really important for Southern California, desert bighorn sheep going across down into Baja. And one of the questions I asked when I arrived here is, what's the genetics of these bighorn sheep right here? So what, does anybody know? And I quickly learned, no, there's been a few little studies, but we don't know what a population of bighorn sheep in Wyoming is. So I just love that question, and so then I collaborated with a number of people in Wyoming Game and Fish, and we started a study. And to do that, I recruited Dr. Sierra Love Stowell, who did her PhD at Boulder on cutthroat trout with similar studies. And so she's addressing these questions. So what is a population of bighorn sheep? What's their population size? What is the impact of their pneumonia outbreaks on their genetics? And so we have a number of questions that we want to address, and that Sierra is answering a number of these already. Wyoming Game and Fish has a number of the ungulate species divided into hunt areas, and each ungulate has a different hunt area. It's a little confusing. You have to learn it for each species. So bighorn sheep have their hunt areas. Does that reflect their genetic biology? So that's one question we have. how many individuals are in a population. Once we define what a genetic population is, how many individuals are there? We can use genetics to determine that. Is the population genetically healthy and stable, or are the genetics indicating fluctuations or inbreeding? What's a sustainable level of harvest? We won't decide that. We'll just give a layer of data to those deciding agencies that will help us. What's the effect of translocation? I'll show you another map a little bit later that bighorn sheep have been moved all over the place over the last 80 years. And so we don't know those sheep that came in from Oregon or that came in from in Montana. Are those genetic lineages still in Wyoming? Is the Wyoming specific lineage still alive? Those are questions that we're addressing. How does sheep move across the landscape? What's the gene flow? So the genetics can help tell us that. Wandering rams are a problem in Wyoming. So there's that pneumonia disease that many of the scientists strongly believe domestic sheep have a factor in. Domestic sheep have certain organisms in their air systems that they live fine with, but when a bighorn sheep goes nose to nose with a domestic sheep, they often contract those pathogens and then can die from it. Rams like to wander. So they have their little ewe group, they do their mating, and then they wander, and sometimes they wander into a domestic sheep area. And so the current policy of Wyoming Game and Fish is if there's a wandering ram that's touched domestic sheep, then they usually have to kill that ram. Now, one thing they want to know is where did that ram come from? Right or wrong on that decision, they send the sample to us because we've just created the databases throughout Wyoming so that we can tell where that wanderer came from. And if there's a disease transmission, did he bring it from his own population or might he have brought it from domestic sheep? As we all know, it's a very hot issue with agriculture and wildlife right now. So data is good to help answer those questions. And where did poached individuals come from? So when the Wyoming Game and Fish Forensic Lab gets a sample, then they can test for that, and we're helping to develop new markers, DNA markers, so they can do that. So we collaborate for this project a lot with hunters. Many of you perhaps have donated samples when you hunt a bighorn sheep, a pronghorn, a mule deer, a moose, which we work on all of them. And we work through hunter check stations. Wyoming Game and Fish has been very helpful at collecting samples for us. Also on telemetry projects. So a number of people work throughout the state to put radio collars on these big ungulate species. And that's really important because genetics is just one layer, and the telemetry information can tell us a lot about what's going on with these populations. So we get a blood sample from those captures. And this is what our samples come. We get almost everything from a wildlife species except the squeak or the honk or the meow or whatever. We work with whatever we can get DNA from. So all kinds of things. So there's papers on that diagram with blood spots. I use them for my hummingbirds. I take a tiny little blood sample and I put it on a paper, and then we analyze this DNA and we test for diseases with it. And some of our lab members that are working in the lab. And so for conservation genetics, I wanted to add one more DNA marker, which is a Y chromosome marker, so that helps us track what the males are doing. I mentioned how mitochondrial DNA is the mother marker. We can test mother lineages down through ancestral time, and how our nuclear DNA, those SNPs and microsatellites, can test our overall genetic diversity and answer questions. So with bighorn sheep, we've used the mitochondrial marker, and it's really important in this species because they exist in ewe groups. It's like a really experienced female who then leads, she's the lead ewe, this band of youngsters and mostly females in her group. And that group may be 15 animals to 50. It varies across their habitat. The rams come in, they live in bachelor herds or individually, but they just come in for mating time and then they leave again. So it's almost like two different species that are having a movement dynamic across a landscape, and that's why we need different markers to tell what's going on. So there's that ram marker, the Y chromosome DNA that we study, and that can help tell us about those wandering rams or how translocations are doing from the male lineage side of it. And then I mentioned microsatellites, how they're really useful for identifying individuals and identifying what a population is. And the genomics, so Sierra has developed terabytes of data, as I mentioned, tons of data that includes over 40,000 of these SNPs, these single nucleotide polymorphisms, that are giving us a fine scale resolution of what bighorn sheep look like in Wyoming as far as their genetic health. And this is color-coded, very early data from Sierra, which is really awesome data, color-coded for a genetic cluster. So it's a map of Wyoming, and each color is referencing a genetic cluster. So you can see down here in Ferris-Semino area, that's the same cluster as Devil's Canyon. And that's due because there were no bighorn sheep there. Wyoming Game and Fish translocated some. They did translocate some from other areas, and we'll be examining whether there's Oregon signature there or some from Montana signature, so that Wyoming Game and Fish and all of us taxpayers know what our bighorn sheep are, what's the composition. And we can see from the GYA and beyond that we have different populations. So the Absarokas are actually kind of two different units here, so a purple unit and the green unit. And it can be different from the Jackson region herds and Whiskey Mountain, Darby Mountains. So this is hot off the press, courtesy of Sierra, which is pretty exciting. It now gives us an idea how these populations are. The next thing she'll look at, she's just starting to look at, is what's the gene flow among these populations. So another thing I mentioned was the translocation history. So this is a map that you can actually get online if you do a search for bighorn sheep, translocation, and it tells you where bighorn sheep, this is the map of Wyoming, where bighorn sheep have come in from other states over the decades. So they've come in from Oregon, as I mentioned, and from other areas. And we want to know, are those lineages still alive? What does that mean for the health of Wyoming bighorn sheep? Does it matter? If there's certain genetic good things or bad things from those other states, it's something we want to know about. Are there factors in the DNA that give more susceptibility or resistance to some of the pneumonia pathogens? And so talking about the disease, this is a bighorn sheep with snotty nose. So it's got a pneumonia disease, and we still don't know the cause. It's multifactorial. We don't know whether mycoplasma is the most important or some of the Pasteurella families. Those are different microbial pathogens. But one thing we do need to know, in addition to what's causing it, is what is a population. Because when we study disease, we have to know what our denominator is. When we study if there's so many incidents of disease in a population, we have to know what the population is. So that's really important, and genetics can help with that. Do diseased or unaffected herds or families differ genetically? That helps management, so they can know what's going on across the landscape. And are inbred individuals more susceptible or less? Sometimes we found that it doesn't matter in other wildlife populations, but for bighorn sheep, we don't know that yet. And are more genetically diverse herds a good thing? With bighorn sheep, they're very adapted to their environment, and there's actually a thing called outbreeding depression, where if you bring in a sheep from another area, say it was a desert bighorn sheep, and brought it into an area it admixed or it bred with others there, it can be bad because its lambing cycle will be off. It is evolved for the desert. It has a lamb or two lambs when it's the right time in the desert. So sometimes genetically diverse is not a good thing. So that's something we'll be studying. And so maybe I hope that you got some information on how many different kinds of information can be important for these species. As an example, for any wildlife species that's at risk for decline, is that we need to know genetics is one layer, but we also work so collaboratively with those experts that know about the habitat, about movement through telemetry studies, about other disease biologists. It's nice to be a veterinarian so I can understand other disease biologists when they talk. The physiology, I mentioned the desert bighorn sheep versus Rocky Mountain bighorn sheep and how they might have a different physiology. The demography, or the number of individuals, their sex composition, what is the actual makeup of a certain population? Are there a lot of older individuals, or are there a lot of younger individuals? And their evolution. All of these mix together to combine for conservation action. And so many, many stakeholders are involved. And so I wanted to give an example of some of the of our collaborations, that we do this for all the species we work on. We're networked in with game and fish agencies, federally and locally, with a lot of NGOs, non-governmental organizations, and conservation researchers, and then also other people at our university and other universities. And so it's not done in a vacuum at all. It really takes a lot of people to do these projects. And just as an example of some of the other projects we work on, they span from mountain lions to sea otters and hummingbirds, and they all have commonalities, but they have specific issues related to their biology, and we adapt our genetic studies to do that. And on the right-hand side of the panel, you can see the team that I'm so honored to work with in my lab. I'm very, very lucky to work with some very brilliant students and researchers. And so what I wanted to close with is what you can do when you think about wildlife conservation. So I'm sure you have your own opinions, but and this group is obviously engaged, but one thing you can do is, just like you've done, is learn about these different tools for wildlife. Because as a voter, you make a big difference. Wildlife is under threat for getting funding. Support your local game and fish agency. They are the ones who are doing the on-the-ground work, applying our data, and they do really an awesome job. It's a tough job. Be a voter, be proactive. And again, I know I'm speaking to the audience that already does that, but I think that's really important. And there are volunteer opportunities. So we do a bit of citizen science with our hummingbird work, and the hunters and other people who are engaged give us sightings, give us samples, so that's really awesome. So I thank you for listening, and I welcome questions.
How do you capture sheep to study them?
So the question is, how do people capture sheep to study them? So we collaborate with the people who capture. In our lab, we capture hummingbirds because I'm a hummingbird bander, but because it costs thousands of dollars and real expertise to capture these animals, we rely on the university researchers and Wyoming Game and Fish to capture them. There's probably a couple different ways that are the main ways. In California, I actually participated in it. One way is the helicopter net gun. How many people have seen pictures of helicopter net guns? A number of people. So the helicopter pilot, who's really expert at it, will fly over bighorn sheep herds and then kind of herd out an individual. An expert net gunner, it's a regular gun with 4 bolts that shoot out with a net. And the net shoots out over the bighorn sheep. And in my earlier days, I flew on the second helicopter, jumped out, usually into cactus in the desert, and then untangled the bighorn sheep with 2 or 3 others. And then we would either fly it back to a base camp where we would put a radio collar on, take a blood sample, check its health, or we would process it right in the field and then let it go again. Another way is drop nets. People can bait in, that experts in Wyoming Game and Fish can bait in bighorn sheep and then have poles that they're there secretly and then let the net down. Or there's drive nets where bighorn sheep or other ungulates can be herded into a net. You can see there's a lot of danger to it. At any one capture, between 2 to 5% can die of the individuals that are captured. So there's some risk. Pronghorn are especially at risk. It can be up to 20% that die in a capture. And that's one reason why in our lab we like to make use of samples that are already collected and hunter samples where hunters go to check stations so that we can minimize how many animals have to be captured because there's risk to people and the animals.
Are emerging types of genetic engineering technology like CRISPR being contemplated as a means of wildlife conservation? And if so, what do you think about it?
So the question is CRISPR, I'll explain that in a minute, being contemplated for wildlife conservation? So CRISPR is a technology, it's an acronym, and don't ask me to define it. Google it and you'll find out. But they can cut out bits of DNA and then put new DNA in it. And so people are contemplating it. It's a social decision, an ethical decision. For instance, there's some researchers that want to bring back the marsupial Tasmanian wolf that's extinct. And there's samples from museums where they propose to use CRISPR to insert that Tasmanian wolf DNA from ancient times into a receptor animal. Similar to Dolly, who was the first genetically engineered domestic sheep. That wasn't CRISPR that was used, that's more recent technology. people are using it. People in my field are doing a lot of communication about what we think about it. Ultimately, it's going to be a societal issue. But I think people like us geneticists can tell you what's the risk, what's the benefit. People have talked about bringing back mammoths. If the mammoth doesn't have a habitat, what's the mammoth going to do? So I think there's more than just thinking about the animal. Is what's the habitat? What would that mammoth carry as far as diseases that might get transmitted to our native animals? It's a hot issue. Thanks for bringing it up. In the back, the far corner.
Yes, I understand that in the Bighorn sheep units that I think what we had up there is a Jackson Hunt unit. There's also a sub-unit of that, which is the Teton sheep, which I think are in kind of trouble population-wise. And I also understand that it's kind of genetically distinct, and that they intermingle with the Jackson herd, which is in fine shape, but that they don't interbreed. And if genetic diversity is fundamental to a herd's longevity, why would that happen? Why would they not try to survive their own species?
So tell me if I'm reiterating your question correctly. So you mentioned some herds that are in this area. One's in trouble, one's not. They seem to intermingle. And is that your question? And why?
They intermingle, but they don't mate. In other words, they're two genetically diverse groups.
Right.
Okay, and it would be to the advantage for survival of the Teton herd to mate with the greater Jackson herd.
Okay.
But they're not doing it.
Right.
It's to the detriment of their survival. And I don't understand why that would happen.
So why over ecological time or evolutionary time are these bighorn sheep herds that are adjacent to each other not mating? You know, there's a lot we don't know. As I mentioned in my first sentence, these wildlife live secretive lives. So in each of those herds, they may have disease issues that are different. We know that across the state. So the pneumonia story is different between herds. It could be that there's a certain U group that has developed within those various herds that's different from the U group in the other herd. And maybe socially they don't want to meet. Maybe we just haven't experienced it over evolutionary time, where, I'll bring up another term, metapopulation. That's a group of populations where those two herds that you mentioned are staying separate for now, even though they're adjacent. But give another set of decades, they will intermingle. We live on short time spans. So our view is pretty narrow, like our previous speaker told us, is that maybe they just haven't had the chance yet. I don't know the answer. We'll probably find out more answers over the next year. Yes?
On your map that you had up there earlier on the different population segments in Wyoming, there was a gap, at least from back here in the bleacher section, around Grand Teton Park, which sort of relates to the previous question, but also clearly Yellowstone was not included in your database. Can we assume that they're the same populations represented in those different colors? And have you looked at any of the paleo data to see what some of the origins may be of these populations? And I'll make one comment on this last question. I think we have to look at the landscape barriers that are between those two populations, why they're not getting together with the development highways and fences. So there's a couple of questions, one response.
So one question was on the gap on the map that I showed. So one is that we just haven't had samples from Yellowstone. So Yellowstone is its own political entity, and so that'll be a piece that we'll want to fill in at some point. It's like its own state, as many of you know. So getting samples requires a lot of permits and collaboration, and that's something we work on, but we have to do it step by step. Another thing is there's other gaps that you may have noticed, and it's probably just because we haven't had samples. Maybe there isn't hunter samples available in that area or telemetry studies. And then the other is there's gaps because there's not bighorn sheep habitat. Let's see, you mentioned some other questions, if you could remind me.
Well, have you had a chance to look at any of the paleo remains, just look at origins, if there was isolation or, patterns?
So that's what we're starting to look at. This is just Sierra did this a couple weeks ago, so we're, just in the early stages. So I think stay tuned. Yes.
How are the hummingbirds in Wyoming doing?
So that's a project of my grad student Brady Godwin, and so he and I have banded the last couple years. He's just extracting the DNA. He's sent the DNA off for the whole genome analysis, but he hasn't had a chance to analyze it yet. So it'll be another year or so. He has worked on some hummingbirds and their genetics in California, and it's really looking interesting where we thought it would be panmixic or all interbreeding, but it isn't. So we might guess that in Wyoming, it's like a sky island kind of pattern where our little mountaintop hummingbird populations, broadtail hummingbird, for example, will be separate genetic units. We're going to test that. We don't know yet. One more?
Yeah, you followed West Nile viruses come across, and have you been looking at chronic wasting disease? And I realize it's a prion disease, but how much cross-species movement is there of that specifically?
So the question was about chronic wasting disease and associated questions. And that is such an interesting disease. I am working with my PhD student, Melanie LaCava, and she is studying the genomics of mule deer throughout the state and also pronghorn. Pronghorn don't get chronic wasting disease, but mule deer, elk, and other species do. And we're also looking in the mule deer and in the future, starting next year, elk, to look at the genomics of a susceptibility gene in elk and deer that gives some kind of resistance or susceptibility. It's actually how fast disease progresses in those species. And so what we'll be doing is creating a statewide Wyoming layer of their genetics, what their populations are, of their disease susceptibility, and then looking at that front of chronic wasting disease as it spreads kind of north and west towards the Continental Divide. And what's going to be really interesting from my point and from Wyoming Game and Fish and many of you is brucellosis is up here, right? And then its front is kind of creeping down, the chronic wasting front. There's one in Pinedale that just got identified, a deer that's had chronic wasting disease. What's going to happen when those two disease fronts merge? That'll be really interesting. So yes, the prion diseases are hot on our mind. Thank you.